In each, there should be only one tiller, one plant per hole. So when you're coming to do your crossing, like this, you should be able to remove these excess tillers and only leave one plant standing. So for this normal standards, for you to ensure that your work is efficient. So apart from those, there are insects, pests, which Professor will want to talk about, especially on these. So you can come close at least.
They did do stalk borer control, but we still have evidence of the larvae having been there. But I think when they controlled, it did not continue. So you did not get dead hearts, but you got the leaf damage. So this maize can still survive because it's only the small leaf damage that managed. And we have another one here which has the same pattern. And you remember when we were talking on pest control, pest and pest control, the difference between the fall armyworm that is now devastating maize in Africa is that they don't do this. They eat in a manner that is not in a pattern. So it's very easy to tell the damage from the one of the stalk borer, between the stalk borer and the fall armyworm.
Okay, you remember the isolation distance for maize? Yes. Stem applies 400 meters for basic and then 200 meters for certified seed. But this is for demonstration purposes. Okay. So we head to.
I have a question.
Yes.
Yeah. Concerning like the pest, like this stalk borer. Like in my country, I can see even if it start like this, then at the end even it will consume the whole fresh leaves. Then at times even the plant cannot grow again.
Yes, they start like this because you remember I said it starts eating from the heart. So this was eaten when it was still small inside there. But I think they managed to put the control in time, and that is why these other ones are not coming out with the same. So the larvae that was there that managed this damage actually died. But if it didn't die, then it was going to continue eating, and you would either get the dead hearts, or if it survived on, then it goes down into the stem, and you saw the open stem that we had. So that is what happened. You can stop the progression if you manage to put the control in good time. But the best thing is actually to prevent so that you don't even get this.
What time of do you till? Like, okay, removing the other one? Because like if you leave up to this time, I think it will disturb the roots of the other one if you remove it right now.
Yeah, actually, for hybrids, normal maize cannot tiller. Yeah? But for hybrids, because they are very, I told you they have what we call what? What do they have? Hybrid vigor. And that's why they tiller a lot. This is not a normal maize. These are basically tillers, and they need to be removed very early. As they grow, they need to be removed. But during planting, most time you plant 2 seeds per hill. This is a hill. You plant 2 seeds per hill. So you have to thin that hill to just one seedling. You have to leave only one plant per that hill. Yeah, so that you reduce on competition. But this is a tiller, and the reason for the tillering is because the plant is showing heterosis or hybrid figure. And you have to take, because this one at the end of the day, you not get anything from there. Or you are going to get very funny cobs. Yeah? Yeah.
Now we cross maize. Sindio? The difference between maize and beans, like we said, common beans, but because there are some other beans which are cross-pollinated. Common beans are self-pollinating at about 95%. But for maize, it's a cross-pollinated plant, and so the approach would be different. The male plant and the female plant equally have to be planted at different times. The male plant has to be planted earlier because the male part of the plant is the tassels. It is in this that we have the pollen grains to use. And it is the ears that give us the female parts that we are going to pollinate. Now the female parts just when you have planted your crop, you wait till when it just starts showing the silks. This is what we call the silks. Then you cover these silks with a transparent polythene sleeve, but that is open at one side, like so. You hold this for me. The reason of covering is to prevent pollination. So, after you've covered, you ensure you don't close the lower part because you have to leave this place well aerated. Then, when you come to getting the male part of it, you get a manila bag, like this one. Maybe like this one. So if you put when you put this inside and you shake, on shaking, inside here we shall have the pollen, which we shall transfer to this female part that is receptive. So we cover like so. So this, the seeds we shall obtain here are the seeds that have a mixture of traits. That is the trait of the male and the trait of the female. But the assumption we have, our expectation is that much of the male traits that we are looking for will be expressed in the maize kernels that we shall harvest. But since maize has a lot of kernels, and its traits are quantitative rich control, it has many traits many genes controlling one trait. So you will have to go through selection. You plant this and you check for the trait you want. Before, when you select your female, you also ensure that the female you have got, you remove the tassels so that it's only pollinated. You reduce the chances of having any other source of pollen accessing your ears. The female part is also called the ear. Am I clear? Yeah. Do we have something to be clarified?
One, I'm talking on maintenance.
Yes.
When you are maintaining
yes.
variety, isn't it?
Yes.
You may cover this one and this one.
Yes.
So how can you know that this is the right time to
cover?
To get a pollen from here and
when you get refilled, just after the plants have developed the tassels, after a week or so, if you shake it, you will see some pollen coming out. That is the right time to collect the pollen. But immediately the silks appear, cover them. Because if you give them some extra time, they will easily be pollinated by other sources of pollen, which may not necessarily be from your field. It can be even from a different far distance. Because I'm told pollen can travel some reasonable kilometers. Pollen can come from even 100 kilometers away and pollinate a plant here. So for us to be safe, you cover immediately, you find the silks come. But you ensure that the bottom part of the sleeve is open. And you collect using a manila paper, because this manila paper is able to provide some aeration that is very important. Like I told you, the male parts of this of the plant systems are so weak. So the pollen is so weak. If you subject it to some stress, of course you will transfer pollen, but it will not be effective. And it's so advised that it is done in the morning or in the evening. Am I clear there?
Yeah. Pollen to female part.
Yes.
And you find that when harvesting,
yes,
the cob, the grain are scattered on that cob.
Yes.
What happens to
the
that?
grain is not, the cob is not having a uniform distribution of grain.
Yes.
What happens, all cobs have the female part of that grain intact. It only swells up to become a kernel when the male, when it has been fertilized. Are we clear there? So, if it was not fertilized, it will not grow to become a grain or that seed that you are looking for. So, if what will make it not, what will make your cob not to have that even distribution of seeds is because it was not fully, not all the kernels that were there were fully fertilized. So lack of fertilization, that is even that brings up that.
But I also have an issue here. We have the challenge of the MLN. In this field, we try to control the MLN, and as you can see from him here, this one. It's actually here. We still have MLN affecting the crop. But one thing that has been discovered with the issue of MLN, especially in East Africa, we have grains forming, very scarce grain forming, and then the rest of the crop is empty. And partly it has been associated with the maize beetle necrosis disease. Okay?
Maybe you never get you handle it roughly, where it never survive. What will be the impact on the cob of the female?
Of course, if you don't put pollen, you get no grain. Yeah, you get no grain. If you don't put enough pollen, you also get a poor cob with not enough grain. But anyway, before you even cross, you have to, because you pollinate when it's a bit young. Yeah. So one thing, most of them may not have really exposed the silks. So what you do is you open up and cut. Yeah, like you cut. In case you don't see the silk, you have to open up and cut and keep it ready for crossing. When you get the pollen, you come and just pour it and cover your cob.
Maybe just to add on that about the contamination, when you're transferring your pollen from the tassel, do not put your fingers or inside on the pollen, because that will lead into contamination. And then the other thing that baby was not mentioned is that labeling is so important. You have to label your lines. You label your line, and if you are doing a single cross, if you're doing just a self-pollinated, you just correct, and we are taking pollen from the same plant and then to the same. You have to label that clearly. And when you are transferring, maybe you want to cross from another line to this other line. Usually you label with the female first, and then the male later.
Okay. A question. According to DUS, how do you judge this field in terms of this thing, and then uniformity, then stability? According to what I learned, it's like there's difference in the color of the silks. So I don't know. How do you judge this field?
But this is an OPV, yeah? This is not really like anything so unique. Huh? This is an OPV, so it's bound to go through outcrossing. Yeah? Yeah. But for a real variety, for a real hybrid, you need to see some, like if you go into the farmers' fields that are really working on maize, you see the things are uniform, the tassels are like similar, you see a lot of uniformity. But for this one, which is really an OPV, you don't expect much of it to be like uniform. Yeah. There are so many gene combinations. You understand the breeding? Yeah, there's a lot of gene combinations all over. Tall ones.
They can also come in.
Yeah.
Because I once said that it's 24 hours.
Not more than that.
Not more than 24
Yeah, within 24 hours. Actually, the more you get to 24 hours, the closer you get to 24 hours, the less effective it becomes. Yeah.
So my question is, when you transfer the pollen from the tassel to the silk,
Yes.
how sure are you of what population of the ear you have actually covered? At what stage are you supposed to, how do you identify when to cover the ear? You get my point?
Yeah, I think we need to
Maybe
move
we
like
move
now
to the-
this one, assuming we want to pollinate. This one is a good one. You know this one, because you cannot see the silks.
Yeah.
And at least younger than this. And what you do is, I don't know whether there's one which is really hidden. Yeah. But there are some which are actually like hidden. So you don't wait for it to ever have a here and here. This one, the silks shooting out. But at this stage, what you do is you open up. Just hold for me this one. You just open up this one. You know, if we had something to cut, you just open up. And then you're going to expose some silks. Yeah, you cut, you open, you expose some silks, and then you put the silk bags, the polythene, these ones, the silk bags. You just put them. Assuming we have cut and we have got access to the silk, yeah. Then you just put it there. Yes, and then what you do, you put a tag. Yeah, you make sure that this plant is already tagged, because you don't want to come and do pollination in the wrong plant. And then if you are doing cross pollination, you go ahead and identify where do you have pollen, where do you want your male to come from. Then you put your this is a pollination bag. You put your bag on the tassel, and then you collect enough pollen. And then when you come, already what you do as you collect the pollen, you already label your bag. This pollen is meant for this particular mother plant. So you label your pollination bag. When you come and pollinate, this pollination bag already has a label on it with the mother plant. You come and you pour your pollen.